Okay, welcome everyone to this Friday night webinar. I guess we can call it a happy hour. So now more than ever, there is a huge need for new tools to manage varroa. So really over the past, 10, 20 years or so, beekeepers have been experiencing extremely high losses, 61%. And one of the major reasons for recent losses is Amitraz resistance. Another difficulty with beekeeping in Texas is also the heat restrictions to, thymol, formic acid, and other treatments along those lines. And so one thing I'm really excited about is this brand new treatment called Norroa. And I'm going to bring Adam on the stage to talk about Norroa a little bit. But before you begin, do you want to kind of introduce yourself and give everybody a little background about yourself?
Yeah. So thanks for having me, Garrett. My name is Adam Paschal. I live in North Dakota. I'm the North American Technical Manager for Bee Health at Greenlight Biosciences. We are the maker of Norroa. Like I said, I live in North Dakota. I've lived up here my whole life, so I am from the land of Garrett Slater. Live in Mandan, North Dakota, which is kind of in the central part of the state, but ties all over the western part of the state where I grew up, but I've lived in the eastern part for 8 years. Before I came to Greenlight, I was apiary inspector in North Dakota for 6 years. So I've spent a lot of time working with hobbyists, as well as some of the biggest commercial beekeepers in the country, working with their operations, trying to solve problems with them, as well as just doing health inspections as needed for them to go to other states like Florida and Texas, because they're required health inspections to travel to other states. And I have bees of my own. I didn't when I was an inspector. If you don't know, North Dakota has about a little over 800,000 colonies in the summer. So I was really busy with that. Didn't have time for my own bees. But now that I'm doing this, I have a couple colonies that I manage with my 8-year-old son, teaching him to be a beekeeper. I also help with our science museum and our local zoo to manage a couple colonies for education, as well as just working with Norroa and helping to bring this to market, and help beekeepers learn how to use it, learn what it's about. And I just really enjoy working with beekeepers all over the country.
Yeah, and I think we first met in, I guess, California officially. I think we were sampling one of the same beekeepers and we ran into each other.
Yeah, we met really briefly, I think at the North Dakota State meeting one year. You were with BIP doing a presentation. We just kind of said hi, and then I was working on a project with a lab that's here in North Dakota. You were with BIP, and we ran into each other. It was a rainy day out in the almond orchards,
Yep.
and yeah, two North Dakota boys running into each other in California.
Exactly.
Bees.
But yeah, I mean, you as your background as a state inspector, you kind of realized just how big of a problem varroa is, and maybe that's one reason that you took the step to this company that, and I think they've been developing for a very long time, or at least the other company was.
Yeah, and that you're exactly right. I always said, and I'm still surprised. I planned on retiring from being an inspector. I loved that job. I loved working with beekeepers. But when this opportunity came up, and I learned more about the product, learned more about the company and what they're trying to develop, it kind of made sense as the next step for me to be able to help beekeepers, not just in North Dakota, but on a national level. Help with varroa mites. And just the technology really was exciting to me, and seemed like something that I wanted to be a part of, to help beekeepers kind of take that next step and hopefully be able to get ahead of things for the first time.
Mm-hmm.
So,
Well, perfect. Well, yeah. So Adam's going to give about a 20-minute presentation. If you have any questions, please leave it in the chat, whatever platform you're on. And then after his presentation, he'll answer any questions. So I think his presentation will be about 20 minutes, but the rest will be really safe for everybody that has questions about the new product.
all right, I'm just getting it pulled up here. All right, can you see it? Is my screen visible? I can't hear you, Garrett, so I hope it's visible. If it isn't,
It is. I took myself off the
okay, perfect. I was gonna say, if not, send me a text, because yeah. All right. So, like I said, Norroa, it's a novel approach to varroa mite control. Brand new. Pretty much everything.
Do you want to put it on the presenter mode?
Say that again.
Do you want to put the presentation on presenter mode?
Is it not on presenter mode? It's on presenter mode on my screen.
If you go to the icon on the bottom,
I had it on presenter mode.
weird. Press that again. Weird.
How about now?
No, it still shows you slide and all the slides on the side.
Oh, weird.
But otherwise, it's not a big deal.
Let me try. Let me try something here.
Okay. While you get that, yeah, Deborah asked, will we get a copy of this presentation afterwards? Yes, this will all be posted on our YouTube page. So whether you're attending live or you might watch recordings later, it'll be on our YouTube page.
I'm trying to figure out how I can do this. Is it in presenter mode now?
It's not, but
That's weird. Sorry, we're trying to learn a new platform here.
so it says presenter mode on your side. So
Now
VK says
now
open
it's not,
mode.
but now
Okay.
I have it in presenter mode.
Yeah, VK says open your presenter mode, then Alt tab, and then share your presenter screen.
Open my, is it in present? Can you still see my screen?
I can, yeah.
Okay, so open in presenter mode. Thing is, if I go to presenter mode, I can't see the screen to share my screen again. I'm not the most technologically advanced here, so.
Yeah, so VK says stop share first and then try it.
Yeah.
And if not, it'll
go into
be fine.
presenter mode and then.
And then VK says go to presenter mode, then Alt tab.
Alt tab. That just made it exit presenter mode. Alt tab. Okay. Hold on, we're doing something here.
Okay, while you figure that out, Lauren asked, what is the YouTube page named? It's called @TAMU_Bees, so TAMU Bees.
How's this look?
There you go. Perfect.
Oh,
Thank
man.
you, VK.
I learned something tonight. Look at that. I'll probably not remember it again, but it worked for tonight.
Hey,
all right.
It only took us a few minutes, so.
Yeah. Okay. We'll try this again. As I said, Norroa, a brand new varroa mite control, is EPA registered on September 25th. Unfortunately, it is not yet registered in Texas. So after the federal registration, we need to go get registrations in every state. We are expecting Texas any day. I'm kind of surprised we haven't gotten it again. It pains me to say that because for a long time we were saying we expected EPA registration any day. And that took a long time, but the states have been coming very quickly. I'm not sure where it's stuck in Texas, but yeah, any day now we expect Texas to come online and for it to be available. I'm hoping it's before the state meeting next week because we will have a booth there. So that's something to keep an eye out for. A disclaimer that we have to have, I'm going to gloss over it because I don't know why we still have to have it. But basically this is for educational purposes. And everything that I'm saying is to the best of my knowledge right now. We're a very data-based company, so any claims that we make, we have data behind it. A little bit of background on Greenlight Biosciences. A lot of our products are RNA-based. That's where we came in with things like Norroa. As Garrett said, it's been in development for a while. We are the third company to work on it. It was created initially by a company called B-Logics in 2010, I believe. They then sold it after a year or two to Bayer Monsanto, who worked on it and kind of sat on it for quite a while. They did do a little bit of development, but the struggle was always to produce the RNA that it's based on at an affordable level. When they were working out, they were thinking it was going to be at least $200 per dose. But Greenlight developed a way to produce RNA at an economical level and really bring that cost down. And so we are working on a number of products. This is actually the second product that we have gotten EPA registered. The first one was for a potato pest called Colorado potato beetle. And we have a lot of other things in the pipeline that we are working on. And that's kind of what we specialize in. So that brings me to Norroa. Like I said, novel mode of action. It is long lasting. I'll get into all this, but it's the longest lasting product on the market for beekeepers for varroa. With that, it supports bee health. It is really the easiest product to use, I think. And because it's RNA based, it's environmentally friendly. The way it's delivered, the RNA, the active ingredient is called Vatiscana. That is the name of the RNA segment that's used. It's double-stranded RNA that comes in a sugar syrup. It's already pre-mixed, pre-measured. There's nothing that the beekeeper needs to do. And it comes in a pouch. You can see with the pouches there, or the picture on the left, there's my laser pointer. You can see in these, all you have to do is peel this sticker back. It exposes perforations that are already there that allows the bees, the worker bees, to access the syrup. You just peel the sticker back, place them either on your bottom boards or on top of frames, and the workers can take that and they store that syrup with the Vatiscana, the active ingredient, in the brood nest. That's important because that once it's incorporated into those stores, it becomes part of the brood food. It gets used for brood food, and that's how the mites get exposed to it. We're utilizing a lot of natural processes to our advantage. You can see kind of on the diagram on the right, we're inserting it into the natural life cycle of both the bees, the reproductive cycle of the bees and the varroa mites. So the queen would lay the egg will hatch, all that development will happen normally. Now what happens is when the mite hides underneath that larva just before the cell gets capped, during that last larval feeding, when the varroa mite is hiding under there, the bees will use those stores that contain Norroa to feed that larva, and the mites hiding underneath will come in contact with it. They get kind of submerged because they're hiding in that cell. Mites have this really cool, unique ability or part of their biology that when they come in contact with RNA, they can just absorb it. There's a few studies that have been done now. We're just waiting to see which university or which entity is the first that will be able to get it published. There's 2 universities and USDA that have worked on it. I'm trying to remember if it was 3 universities, but I think it was just 2. So it's kind of a race to see who gets it published first. But they absorb it, and then it starts a process called RNA interference. This is a natural process. It's how our cells defend against viruses. It's also how cells just regulate themselves. It's how they turn on and turn off messages from our genes and tell our bodies which proteins to make and when to stop making them. In this instance, we are telling the varroa mites to stop making a specific protein. Without that protein, they can no longer lay eggs. So we're in essence sterilizing varroa mites. Because of that, this is not a knockdown miticide. So that's another one of the new things about it. Every product that we have is a knockdown, where we wait for our mite levels to build high enough to where it's unacceptable, reaches an economic threshold where they're doing enough damage, and then we knock them back. With Norroa, we prevent that from happening. You treat when your mite levels are low, and you prevent that population buildup from ever happening. So it's kind of a new angle. It's the first truly preventative product on the market for varroa mites, and it's the first product that was designed specifically to target varroa mites. Everything else is a compound or something like that we discovered works on varroa mites and doesn't kill bees. This one was designed specifically for varroa mites. There has been some outside work done on this. The first 2 papers that have been published came out of Victoria University of Wellington in New Zealand. They have more on the way. There's also 3 universities now in the US. I always feel like I'm forgetting some, but 3 that are working on work to be published right now at various points in the work. But this is from the first paper that Victoria University got published, specifically by Rose McGruddy with her PhD work. What she did, or what they did, was develop what they called mini hives. They're essentially modified nukes. On one side, as you see, was all for brood rearing, so comb where the queen could lay, rear brood. And the other side was essentially for feeding. And what they would do was they would let the queen rear brood, and they would feed Vatiscana. In the publication, it simply refers to as Vatiscana. The trade name Norroa was not picked yet. And they would insert varroa mites a few days before the cell was capped. You see here the normal reproductive cycle of bees and mites and how they overlap. Any beekeeper that has been around for a little while has probably seen this before. The varroa mite goes in after the cell gets capped. The first cell, or the first egg that is laid, is a male. The subsequent ones that are laid after that are female. Mites are obligate inbreeders, so those females reproduce with their brother. If it is a worker cell, there's likely two female mites that reach adulthood and that are viable to leave the cell and reproduce or go off and lay more eggs. And that's how the mite population builds. What they did was on day 12, after the cells were capped, they uncapped them and looked for viable offspring. And what they found was that in cells that were treated or that where mites were exposed with Norroa, that just didn't occur. There was essentially no offspring. There were very rarely were there eggs, and if there were eggs, they didn't hatch, or the offspring wasn't viable. So that showed that it worked. It was doing the job that it was meant to do. They were testing a few different concentrations. It was 2 grams, 4 grams, and 8 grams per liter. There was some application problems. They made some mistakes with the 4 grams per liter, so they had to throw that data out. But with 2 grams per liter and 8 grams per liter, you can see that there was no offspring in any of the infested cells. That's compared to a dsRNA control, so a double-stranded RNA that was just not targeted at anything, and a sugar syrup control, where we saw the average of two offspring or viable offspring in the infested cells, which is what we would normally see. Interestingly enough, the 4 grams per liter is what the dosing that ultimately was landed on. So that's what we saw. So whether it was too low or too high, it worked the same. What's also important with that is that with the 2 grams per liter, 8 grams per liter, so too low or too high, the control, both controls, there was no impact on the honeybee pupae. So that also shows that no matter what they were given, there was no impact. It was safe for the honeybees, which is also very important because you don't want to give any treatments that are going to kill your bees. With that knowledge in hand, we did field trials. We've been doing field trials actually before that work was done for over 5 years now. It's been in thousands of colonies, not just in the US, but in Canada, in Spain, and New Zealand. But this is data from just one trial in the US because it's the most robust trial that we did that incorporated the most colonies across the most geography. So it was in Idaho, North Dakota, Maine, Florida, and Georgia, and over 1,000 colonies. The red line on top is the untreated control, which is not a surprise. If you don't treat your mites, they're gonna grow, they're gonna get out of hand. The middle line is what we call the chemical spring or the chemical control, just the leading chemical. It was apivar strips. I'll just say that. And then the bottom line was treated with Norroa in the spring. We also said chemical fall. So the chemical treatment and the Norroa treatment did receive apivar strips in the fall at 18 weeks. And with the apivar strips, they were put in, everything was started at week 0, so it was roughly May. The colonies started as splits, so they were in singles. That let the mite levels be, relatively low. There was a broodless period. And then the apivar strips worked fairly well for 6 weeks while they were in the colonies, which isn't much of a surprise. And then as soon as they came out, the mite levels started building, and they built about how we would expect to see. With the Norroa, it was applied, and there was no mite population buildup for 12 weeks. And then we started to see a slow increase until the 18-week point. It finally crossed the 3% threshold. So that was 18 weeks, 4.5 months until it crossed 3% threshold. In this specific study, as I said, then both of these two groups were treated with apivar strips and then left to go for another 18 weeks, at which point they were checked to see the survival. And that's really telling, because the only difference in those two groups was the difference in spring treatment. So the Norroa in the spring had nearly twice the winter survival rate going into February was when we stopped tracking them. Obviously, untreated control, you don't treat your mites, you're going to have very low survival. I'm surprised that they even had 15% survival for absolutely no mite control. Again, commercial colonies. Apivar spring and fall had about 45%. Norroa in the spring and Apivar in the fall had between 70 and 75% survival. So the only difference between these two was a spring treatment of Norroa, and the winter survival nearly doubled. So last year I started getting really curious and thinking for fall applications, especially in warmer climates, or commercial beekeepers up here in North Dakota that are getting ready to leave. Sometimes you come out of your honey flow, you're in August or September up here, or just in different areas of the country, and your mites might be higher than you would want to treat with Norroa, but you may not want to wait until those mite treatments are done before you put it in. You might want to get a head start. So I started wondering, is it safe to do a co-treatment and put a knockdown and Norroa in at the same time? And so that's exactly what we did. This was in Florida last fall. Again, untreated control, mite levels went up, and then we decided at 15% mite loads to pull them out so the beekeeper could treat them and try and save the colonies. I don't like to just kill colonies for no reason. So we called them dead, but really they were just removed. And then there was the yellow line is Norroa on its own. So they were below 3%. I would have liked to have higher. Feels weird to say I would have liked to have higher mites, but we started this on kind of short notice. I started in August with the company, and we started this trial in October. So I had to pull it together pretty quick. Otherwise, right around 3%. We started Norroa combined with one oxalic acid dribble, Norroa with 3 oxalic acid dribbles, and Norroa with Apivar strips. Not surprisingly, one oxalic acid dribble in Florida in October didn't do a whole lot. The Norroa, for the most part, stopped the mite population growth, but what you will see is that it pretty well mimicked the untreated control. So I think what we did see for growth was pretty much drift coming in, because as soon as we started removing those colonies, that growth rate actually dropped to match that. Everything else, it pretty much showed that the Norroa was stopping the mite population buildup. It was stopping them from reproducing. So that's really reassuring to see that you can put it in with other knockdowns, and it doesn't affect the performance of Norroa, and they both do about what you'd expect them to. Because it was Florida in October, I wasn't able to do that with formic acid, but I am repeating that trial this fall in 2 locations. One of them is Texas, and it just kicked off over the weekend, I believe, and formic acid was included in that. So in about 18 weeks, give me a little more time to analyze the data, I'll have results on that. But I have no reason to think that you can't combine it with formic acid to get a good knockdown if you need, and then let Norroa do its work to keep those mites low. Along with that, because Norroa kept those mite levels low, obviously there's more that goes into it. There's a whole summer worth of management, but we had almost 0 colony loss in any groups that were treated with Norroa. We lost, I think, 2 colonies in the Norroa and Amatraz group, and I just attribute that to sometimes colonies just want to die, because they were, for the most part, healthy-looking colonies, but sometimes you just can't keep them alive, and there's just not much you can do about it. I think any beekeeper understands that one. So this is based on the Honeybee Health Coalition's varroa management guide. A new one will be coming out that they will be doing the chart on their own. So you don't have to go off of our numbers, but really there's a few different groups of mite treatments. There's the synthetics, so taufuvalinate and coumaphos basically don't even, almost no beekeepers can use them anymore because the resistance is so bad. They can be harmful to bees as well. And even Amatraz, as Garrett said, resistance is definitely growing. When you don't have resistance, it can definitely be effective, but that's definitely something to think about. When you have your acids, there's formic acid. It can be very effective. Every once in a while you hear about maybe a bad batch or something. I don't know about that. I've had good luck with formic. But you definitely need to be considerate of the temperatures, especially down in a place like Texas, or it can be very harmful to the bees. Oxalic acid is definitely effective, no resistance, really not harmful to the bees. Sometimes with dribbling it can be, but it's very labor intensive. It takes, especially when you're vaporizing it, it takes multiple trips. You need PPE, or it can be very dangerous for the beekeepers. So there's definitely the drawbacks for that. Hops, beta acids, hop guard. I've never used it, but I know a lot of people say it's not the most effective. There are some temperature things you need to keep in mind with that. So I know it's not the most popular control method. The essential oils, the thymol-based products, definitely temperature restrictions that you need to keep in mind. They can definitely be harmful to bees. So there's some drawbacks to that. So with Norroa, it's very effective. We have a lot of data to back that up. It just works. There's obviously no resistance. It's new, and we don't expect any resistance to pop up anytime soon. I'm not going to say it can't happen. I think with any product, resistance is always a possibility. So it's something to think about. And that's what IPM strategies and rotations are for. And we have lots of data to show it's not harmful to bees. So part of that uniqueness, it has all those benefits, and there's no PPE required to compare to oxalic acid. The biggest drawback is if you have high mite levels, then it's not the product for you. But if you have low mite levels, it'll prevent those high mite levels from building. One question we get a lot is off-target effects, and that's a big concern for something new. And especially with this new mode of action, RNA can be scary to people because the ways that they've been exposed to it in the past several years. But that's something that a lot of work has been done, not just by us, but again, Victoria University of Wellington has a publication out regarding it. One of their grad students looked at wax moths as a surrogate, another organism that would be in the hive that would have a lot of contact with it, both physical touch and ingestion. And they used that as a surrogate and saw that there was absolutely no off-target effects on wax moths. It was, for better or for worse, it was completely safe for wax moths. There's also been a lot of bioinformatics work done where you look at the genomes of other organisms and see if there's overlap, any risks. And so 39 arthropods known to be associated with beehives or that were insects of conservation concerns. So things like monarch butterflies or other, just other butterflies, ladybugs, things like that, that were either keystone species or just, like I said, species of concern were analyzed. When that happened, there were 6 species that had small interfering RNA hits at 21 nucleotide lengths. That is kind of the threshold of where you need to be concerned. You can see the ones here: monarch butterfly, fruit fly, European earwig. I don't know what the Stratiolaelaps and the Tetranicus are, and Tropolaelaps, which I think most people would actually be kind of happy if it worked against that, but we're not terribly confident in that. But monarchs were a big concern. And so what happened was a feeding assay or a feeding trial was done to assess that, because if you're concerned, the best way to do it is to get exposure and not deal in hypotheticals, but to just to get in there and do it. And so they did that. They fed it to monarchs. They had them exposed to it physically, and there was no off-target effects. Once they analyzed it, they looked at the genes. They looked at any effects that could happen. There's a publication out there showing that there was no impacts on the monarch butterfly. So it's safe. It's been shown that anything that could come in contact with it is safe. And even if there was a spill, if some were to get out of the hive, because it is applied in a controlled in-hive environment, if some were to get out of the hive on the ground, the soil microbes break it down very quick. That's just how RNA works. Is it's in general not a stable compound. That's one of the challenges, is to keep it stable in the hive, and we've been able to do that. But once you get it out of the environment that it's designed to be in, it breaks down very quickly. And lastly, like I said, when it comes to beekeeper safety and ease of use, that's one place where we've excelled. Coming in these pouches, it's very easy to apply. You just peel a sticker, set them down, and that's it. There's no PPE required on the label. Because the EPA was required, it says long sleeves, long pants, socks, and shoes. No gloves, no eye protection, no respirator. I actually think it's funny that you're required to, it says long sleeves, but no gloves. You can get it on your hands, but don't get it on your arms. Something changes when you get up above your wrists, apparently. But whatever you keep your bees in is what you wear for Norroa. You can even see here one of the beekeepers that's been in a lot of trials here. He goes barehanded with just a veil, and there's the Norroa pouch right there. So whatever you're comfortable working your bees in is what works. No need to mix or measure. It comes ready to go. It lasts much longer, like I said, 4.5 months, which is much longer than anything else on the market. And then there's no temperature restrictions, which is a big deal in states like Texas and Florida, just anywhere across the South. Any temperature that you can work your bees in, you can pop your lid, you can apply Norroa. Storage temperatures, we say just keep it below 95 degrees long term. If you're out in your pickup for a day and a day or two and it's really hot, that's fine. But if you're going to be storing it for a week or longer, try and keep it below 90 or 95 degrees. And this just kind of compares that. We say the upper limit is 105, the safe working general safe working temperature for bees. Lower limit, about 45, something like that. But really, you can freeze this, and there's no negative impacts. And that's something that we tested. Again, living in North Dakota, I always think about what happens when I have to order something in the winter. It's going to freeze. So we've done freeze thaw tests, and that there's no problems. Not something I think that people in Texas are too concerned about, but it's good to throw out there. But yeah, you can have it in your hot pickup all day long while you're working. Just bring it in at the end of the day and you're fine. And then, yeah, if you can pop your lid, you can apply it. With that, I will take any questions. I'm not going to leave this slide up just so I can exit, but I'll take questions. Let people give you a minute to take my information there. My phone number and email address. You can also just always contact us on our website, norroa.com. We have a contact us button where you can submit it, and those emails, I'm one of the ones that gets every email that comes in there. So you can always send an email in that way, and we're very quick to respond, usually within a day, unless we're traveling to a conference or something, then sometimes it might take an extra day or something. But with that, I'm happy to take any questions.
Okay, perfect. Well, I think you're gonna. Yep. Okay. Yeah. So if you have questions, please leave in the chat. One question I do want to address with Kirk is our relationship is more. I mean, we come with AgriLife Extension, and so our role is to do trials, like unbiased trials with these products. And so, like Adam mentioned, we just started a trial last week. That's the first time I ever worked with a product, and so we're doing a trial with that product alongside other treatments. And then Chris had a few questions about dosage differences between 1 deep box versus 2 deep boxes.
Yep, that's a good question. So a dose is 2 pouches per box. That's what the label says. So if you're treating a single, it's 2 pouches. If you're treating a double, it's 4 pouches. That being said, there's a couple ways to work with that. Like, as I mentioned, in a lot of our trials, we treated them as singles, and then they naturally, after split, and then they naturally built up into a double. So it was a single that was treated with 2 pouches. They naturally built up into a double, and then we still got that 18 weeks of effectiveness. If you're a colony that is already in a double, so your queen might be laying in both boxes, you have stores in 2 boxes, that's when you want to treat with 4 pouches, because your bees are going to dilute it more. There's going to be more area for them to spread it, the stores around, move the stores around. You want even dispersal throughout those boxes. So if you treat 2 boxes with only 2 pouches, it will either get too diluted, or the way it gets dispersed won't be very even, and then you run the risk of a significant number of cells not being treated with Norroa or with Vatiscana, and then you have mites that won't come in contact with it, and then they'll still be able to reproduce, and you just won't get very good effectiveness.
We had a few questions, actually quite a few questions about the expected cost of this treatment.
Yep. So I will say it's exclusively available through Man Lake. We have an exclusive distribution agreement with them. Right now, as of today, it's available to order in 3 pack sizes. So a 2-pack, which will treat 1 box, a 12 pack, and then a 24 pack. Depending on which one you buy, you order, I believe it's anywhere from $17 to $22 a dose. I have to check again if that's exactly what it came out to. So that's what it is as we've been on the market now for 6 weeks. Our biggest priority right now is bringing that cost down. I mean, we know that that's high. It's also giving you the longest coverage for any miticide, and it's again preventing the population buildup, and it's a new technology. But with that, we know that it's high, and that's our top priority right now is bringing those costs down. We have weekly meetings on our progress for our cost reductions and how we can bring those costs down for beekeepers.
A few questions about using it with honey supers.
Yeah, that's a good question. So you can't apply the pouches when supers are on. I shouldn't say you can't, but you don't want to because you want it stored in the brood boxes where it's going to be used for brood food. If you apply with supers on, they're naturally going to move it up into the supers. It's going to be stored there and it won't be used for brood food. That being said, the pouches are generally emptied within about 48 hours. And then you think of it as like any other syrup that you give them. They'll spend a few more days moving that syrup around, moving it around, getting it where they want to, where they're going to store it. And then for the most part, they're done moving around and you can put your supers on. So what we've been saying is a week between pouch application and then being able to put supers on. Because once it's stored, then they're really not going to move it up into the supers. If it gets into the honey, it's safe. We have a tolerance exemption from the EPA. Even though we're not approved for sale in the European Union yet, because we have to go through, it's called the European Medicines Agency. It's a veterinary medicine product in Europe, like all miticides. The European Union itself has decided that this is safe if it's in honey. So there's no need to test it. They're treating it like oxalic acid. So if it winds up in honey, it's completely safe. But it's not effective if it winds up in your supers. So you want to apply the pouches with no supers on, give it about a week, and then after that you can put your supers on.
Yeah, put another question here. This is a few questions people had was how many estimated treatments per year and is retreating?
Yep. So our label says don't use more than twice a year. That is us trying to promote good IPM rotations. There's no negative health impacts, no drawbacks to the colony if you were to use it more than twice a year. But just as a company trying to promote, good practices, good science, that's one of our core missions. We wanted to make sure that provided people are following the label, that they're naturally getting good rotations and kind of force them to use something else to have that rotation. One treatment a year won't be enough. Like I said, it will keep your mites down for 18 weeks, 4.5 months. But you can follow it up. There's actually instructions on the label for how to do a repeat application if you want to. The best time of year to treat, if you were going to do it once, it would be what we call a spring application, and that's very vague because spring is different everywhere. In Texas, it's very different than in North Dakota because here, that would be May, and in Texas, that's very much not. You guys are probably in the middle of a honey flow or almost finishing one there, but it would probably be better for pre-honey flow. Mites are naturally going to be lower than, and then it'll carry you through your whole honey flow when a lot of products can't be used and your mites are starting to build. Another time would just naturally be that April through June, only because then that will let you get into August, September, October with low mite loads. And then your colony will be starting to produce winter bees that are healthier, larger fat bodies, lower viral loads, everything that comes along with that. And then that sets up your winter survival for higher success. I will take that opportunity. I didn't talk about application and I won't spend a lot of time on it, but on the label, if you're doing 2 pouches or 4 pouches, if you're treating doubles, There's a few ways that you can do that. You don't have to put all four in at the same time. And you don't have to put them all in the same place. So you can put one on top, one on the bottom board. You can put them both on the bottom board or all on the bottom board, all on top. You can kind of split them up that way. And you can put them in at different times. So you can do what's called a split application where you half at once, let the bees empty those out, and then come back with the other half. As long as all the pouches are applied within 3 weeks of each other, you can kind of mix and match however you want to. So if you're not comfortable putting all four pouches in at once, for whatever reason, you don't have to. So you can do two at a time. If you're treating doubles, come back a week and a half later or something and do the other two. That works just fine too. So I thought that might be important to touch on.
We had quite a few questions about using this in conjunction with other treatments. This is just kind of one example. Some people asked about using thymol or formic in conjunction, and is that going to be necessary based on the mite levels? Which it sounds like on your presentation that you might have to if your mite levels are too high, that you almost need to knock down in conjunction with this treatment.
So can we? I'm looking at the one that's pinned and what you're asking. I'm not sure if they're the same thing. In a hive with low mite levels, can Norroa be used as a sole agent twice yearly to prevent mite buildup? It certainly can. With that question there, it kind of depends on where you're at and what if you apply with mite levels low, you come back and they're still low, you can absolutely apply Norroa again. And I think about a state like Texas where you probably have brood year round. That might not be enough. You might need to do something for the other, few months. A state like North Dakota where, my bees are going to come out of winter in about April, and this year is really late. I still had brood a couple weeks ago. It was really, I had like one frame of brood a week or two ago, but I still had a little bit. But I really think about it, mite management from about April or May until about October. In that situation, it's absolutely possible to use just Norroa if things work out right. I did have a colony, so because I was with the company and I'm the technical guy that runs trials, I ran a little experiment on one of my colonies, and Norroa was all it got this year, and I'd never washed a single mite. So the other colony that didn't get Norroa, I had to treat with formic acid this fall. So take that for what it's worth on like the smallest trial that you can possibly run.
Yeah, so probably for Texas, I mean, colonies start brooding up sometimes as early as late January, all the way until now. Our colonies are going broodless, but we only have a few months of broodless, probably compared to North Dakota, where
I mean, yeah, I could see a situation where it's possible. I think the key is to make sure you're still doing mite washes. And that's the apiary inspector and me coming out that, even if you're doing something like this, still do a mite wash. And that's to protect you. I mean, there's always the chance that a bad batch could be made, that there could be something else going on. The best way to know that everything is going right is to still go in your bees, do at least one mite wash a month, and just be on top of things. That's just going to make you a better beekeeper in general.
Yeah, and that kind of follows up in your question. We had a few questions about measuring the efficacy of it, right? So is alcohol wash still the most effective way of measuring whether the treatment's working or not?
To me, yeah. I mean, the best way to know that it's working is to check your mites and see if their population is going up. In a state like North Dakota, in the middle of the summer, you might see a slight increase, only because we've got such a really dense commercial population, and not everyone is really good about controlling their mites. So you'll see a little bit from drift, but what we've seen is that those colonies that come in through drift, as long as you still have the Norroa stores in your colony, those mites that come in through drift will get exposed, and then they can't reproduce. So that's really nice. It's kind of an insurance policy a little bit against bad neighbors. But you still need to check your mites. I'm never going to tell people that it's so good that you don't need to check your mites, because to me that's just irresponsible.
Sure. Yeah. So with the question, right? So I guess it's not necessarily killing the mites, you still have mites in your colony, right? And so
Yep.
is alcohol wash still I mean, I suppose slowly over time, those mites will start dropping, right?
Yeah.
So I guess figuring out
Yep.
like when to monitor might shift a little bit versus other treatments that's sort of a contact kill or like a fumigant kill.
Right. You won't see a big mite drop. Um, yeah, you don't see the big mite drop like you traditionally will. So um, the best way to know that it's working is to see that you don't have an increase in your mites.
So almost like continuing monitoring over time, like after the treatment.
Yep.
Because even if you apply it, I guess you might see mites, but they might not be fertile, right?
Right. Yeah. And really, I mean, you know, if you're applying when you have, you know, I think about here, and I always relate things to North Dakota because that's where I am. You know, a lot of times in May, I'll do an alcohol wash and I won't see mites just because I've come out of winter and my mite levels are so low, I won't see anything. The nice thing is I can go with Norroa, I can go into August and then I'll still see that in August, you know, all the way through the honey producing season. Or I might see, you know, I might shake a 1 because I've got within 2 miles, I know I've got 6 different commercial yards, you know, going 2 miles in every direction. Um, but that's way better than I am a couple, you know, even a year ago. That's way better than I would have been.
So one other question we got a few times was effectiveness against AAA laps and whether you guys have investigated that.
We are working on that right now. I don't know if they've been shipped yet or if they are getting ready to ship, but we are sending Vatiscana over to Thailand to test against AAA laps, along with roughly 6. I don't know what the final number was, new sequences, just because we don't know if it will be effective, how effective it will be against AAA laps. And so since we're doing the work, we decided we would invest the money in developing some new targeted sequences against AAA laps that have a higher likelihood of having an effect. If it turns out Vatiscana does work, then great. Then we spent a little bit of time and money on these new sequences that we didn't need to, and we can shelve them and we still have them in our pocket. If it doesn't work, then we have these other ones that we've worked on and we can move forward with. But yes, we are actively in the process of that.
Another question that Chris had was there a difference in treatment effectiveness based on region, like the north or the south, like differences in humidity, temperature?
No, no, there's really no difference on that.
Okay, we just have a few more. This is a question we got from a few people right away about
Mhm.
mRNA and is it similar to COVID? And do you want to explain
Yeah, and
what
that's a really good question. It is not an mRNA solution, but it does work with mRNA. So I'll get into the biology a little bit. So there's a hierarchy of information in biology. So everyone's familiar with DNA. That's where our genetic code is at. RNA is what translates that DNA, reads it, and then it carries the message and tells our bodies which proteins to make. And proteins are how our bodies actually function. Proteins are what does a lot of the heavy lifting. There are a lot of different kinds of RNA that do all that work. We hear RNA and we think it's ubiquitous, but there's a lot of different kinds of RNA. What we were all exposed to, what everyone heard about during COVID, was mRNA. So that's messenger RNA, and that's what carries the message, hence the name messenger RNA. What this is it's just double-stranded RNA. It is not a living strand. It has not come from a living, I guess you could say it comes from a living thing because we utilize yeast, but it doesn't come from yeast. It's actually the only way I know that it's made is with fermentation. But there's no way for this to recombine. There's no way for it to mutate because it's not a living strand of RNA like was used in a lot of the vaccines. What this actually does is all of our bodies have that messenger RNA to carry the messages from our genes, from our DNA. Same with the varroa mites. They have this messenger RNA to read their DNA, carry that message to produce the proteins. The double-stranded RNA it's actually a long strand. When it comes in, there's a mechanism called, or a cellular machinery, they call it, called dicer, that then breaks it up into small pieces, which is, it happens all the time. It happens within our bodies with different things too. And those small pieces then find a complementary piece that matches up. So there's 4 letters that everything's made up of, these nucleotides. It finds the complementary piece that matches up with it, and it binds to that. So it locks in, they match up. What it's matching to is messenger RNA that already exists within the varroa mite. And once that messenger RNA, that mRNA that exists within the varroa mite is bound, it can't do its job anymore. So what we're doing is we're actually blocking the mRNA that exists in the varroa mite. We're going a step beyond. Instead of introducing new mRNA like was done in the vaccines, we're blocking the existing mRNA from doing its job. So we're, like I said, it's kind of a step further, the next step beyond what the vaccines were doing. Instead of introducing new mRNA to do a new job that wasn't being done before, we are stopping the existing mRNA from doing the job that it's supposed to do. Mm-hmm. Because of that, that protein isn't being produced anymore, and that's why the eggs aren't being laid.
Okay, perfect. Well, we just have a few quick hitter questions. I know you have a concert you need to get to.
Oh, I got plenty of time.
Okay. Yep. Yeah. One question was, I guess, registration. So I guess we talked about this kind of offline about registration timeline, not only for Texas, but even for other countries.
Yeah. So like I said, we got registered with the EPA on September 25th. Now every state kind of has their own process. Texas will be, last time I checked, I think our estimated registration date for Texas was November 10th. So really any day now. California was actually really quick because they had a lot of pressure. So they started reviewing it before EPA was finished. So we got our California registration within a week of getting our federal one, which is unheard of. As far as other countries, we are under review in Mexico. We are under review in Canada, in the European Union, and in New Zealand. Each country takes different times. I have no idea how long we expect for Mexico. Canada's probably going to take 2 years. At least they're actually, I'm told, more difficult than the EPA. The European Medicines Agency is probably a year to a year and a half. And I don't know about New Zealand because I haven't followed that one as closely. So I just don't know because I haven't paid much attention to how that one works yet. Each one's different, and each one is a government agency. So you take everything with a grain of salt because there can always be delays and different things. I worked for the government for 10 years. So I'm painfully aware of how all that goes.
Yes, we had this question plus a follow-up question. So yeah, from Jay Dirk, is queen that's fed as a pupa will carry this in her blood? And then Chris, actually he said F-U, I thought it was F-U, but I think it means follow-up. I'm guessing that this is simply carried in the bloodstream of the larva bee, which then gets fed on by the mites. So the RNA does not affect the bee itself. Is that correct?
Okay, so the queen question. The queen will not be fed this as a pupa because this gets incorporated into the brood food. So late stage brood food is a lot of like either sugar syrup or nectar, whatever they have stored in, and pollen. And you think about what queens are fed, that is royal jelly. So the queens will not see this because they don't get exposed to any of the stores that are in the hive. They exclusively are fed royal jelly. So no, the queens will not be fed this, and so this will not be carried onto her brood. There's no transmission of this from bee to bee. We also have tests by multiple entities that we call soaking assays to prove the mechanism of how this works, that varroa mites are absorbing this, where we remove them from the cell, just put a drop of either brood food or Vatisca. Each one has done it a little bit differently, but put a droppa on them and then see if they can reproduce. Where they're basically removed from the brood is removed from the equation. Either see if they can reproduce, or a genetic analysis is done to see if the RNAi process has taken place. And we've seen multiple times where that happens. So they don't need to feed on any sort of larva that has consumed this, but the RNA has still made the dsRNA has made its way into their body. There's one university, it hasn't been published yet. I don't know where it is, when it will be, so I don't want to name them. I don't want to infringe on their work. But they've actually used RNA, double-stranded RNA that had not translucent, that's not the right word. It glows. Garrett, help me think. Fluorescent. There you go. I knew you'd think of the word. A fluorescent dye attached to it, and then they glued a mite to the head of a pin, put a drop on the mite, and then they were actually able to track it into the mite's body. Not only that, they were actually able to observe the RNA interference process. They were able to see like different things firing within the body, I guess is the best way to explain it. So they could kind of see that happening in real time. So it's really cool stuff. I can't wait for that to come out. That's something that they did on their own. So I've only seen bits and pieces of it that they were kind of to share with us. I've asked them if they'd let me use the images, and they just say no. They want to make sure. I mean, I don't blame them. They want to be the first ones to get it out there. But it's really cool stuff. I can't wait for that to come out because it's really cool.
Yeah, and you've probably already answered this question from Chris Grover. Does Norroa interact with other treatments? If you have a spike in mites after treating with Norroa, can you use other products to knock them down?
Yeah, from what we've seen, there's no interactions at all. You can absolutely combine it with a knockdown. From what we've seen, that works great. And if your mites are higher than what you would want to maintain, we actually encourage it. So.
Perfect. Last question. So this is kind of follow up your other question about monitoring the success of the treatment or not. And so James is sort of asking, is a brood assay or like a Harbo assay sort of a more accurate way of measuring whether it's working or not? And have you tried like a Harbo assay or like a brood assay?
Oh, I think we have. I'm trying to think back on all the trials. There have been some where they have, yeah. We generally try not to, just especially in my trials, I don't want to do super labor intensive things. So yeah, there have been some.
Okay, awesome. Well, that was the last question. Do you want to put up that last slide again in case? I guess I was put your email in the chat too.
Yes, and some let me get that going so I can do this right.
Sure.
Oh man, let's start at the beginning of the slideshow. Hold on.
And some beekeepers had questions about the papers you include in your presentation. Are you willing to share those papers, and then I can just include that?
Yes. So actually, if you go to our website on norroa.com, there's a resources tab, and all of the publications are available to be downloaded there, as well as on the main page you can get like the product label, the SDS sheet, and we're going to move those to the resources tab. We're in the process of kind of redoing the website so that it's better organized. But yeah, both of the papers, the McGruddy and the Bulgalera, which was the one about off target or lack thereof, were both included there. Can you see, did the slide share?
I'm not seeing it.
Let me try again. Oh, no, it exited. Stop. I don't know what I'm doing. I'm trying. Here, I'll just do it this way.
Yeah, I this video will be sent to everybody, so I can always include your information with that also.
Yeah. Give me 1 second here. I'll get it. get it going. Again, I am not a tech person, so.
Chris asked where he can get one of those cool hats that you're wearing.
If you are at the Texas Beekeepers Association meeting next week, I will not be at the booth because I will be at the California Beekeepers Association. But Eric Walgren, who you can see right here, he will be there, and I know he will have some hats. So, or I think he'll have some hats. He's supposed to. So swing on by, say hey, let him know that you were logged into the webinar, and just ask, and I'm sure he'll give you one.
Is there like a discount code, or you just go up and he'll give you one away?
Just say, hey, can I have a hat?
Perfect.
I'm really excited about Norroa. Can I get a hat?
Okay, awesome. Well, Adam, thank you for joining us today. There'll be a lot of people that are joining in, probably watching the recording, but yeah, really appreciate it. And like I said, his email's right there. If you forget his email, just shoot me an email and I'll try to answer any questions.
Yeah, and like I said, even if you forget that or you didn't get it, you can go on our website, contact us through there. And Garrett has my email too. So Garrett, you can give it to anyone that asks for it.
Okay, perfect. Well, sounds good. Thank you.
Yeah, thank you for having me.